Welcome and thank you for joining us for the second Agro Lunch and Learn webinar of 2023. This webinar series is possible through the generous support of Eurofins AgroScience Services. The Agro Division thanks Eurofins very much for their continuing sponsorship. My name is Solito Sumulong, Regulatory Director of AgriThority and Board Member of the ISAA Agrochemical Network, and on the Executive Committee of the Agro Division of the American Chemical Society. I'll be serving as moderator for today's webinar. You should use the Q&A tool to submit questions and comments, which will be monitored and answered during the presentation. Before I introduce our speaker, I'll highlight the enormous benefits of becoming an Agro member. Agro enhances your science, builds your scientific network, and supports your professional development. Please go to agrodiv.org and click on Become a Member at the very top of the page to become an affiliate for $20 a year, regardless of the membership status with the American Chemical Society. For the American Chemical Society fall meeting, we have excellent Agro Division programming, as well as the rescheduled 50th birthday party of the Agro Division, postponed since the global pandemic of 2020. The abstract deadline has passed, and the organizing committee is putting together the session schedule as I speak. If you have any questions about the programming, contact Aaron Gross of Virginia Tech, our program chair for San Francisco. Now it is my pleasure to introduce our speaker. As Global Director, Science and Technology for AgriThority, Ignacio Colonna has technical focus on incorporating methodologies, equipment, analytics, and other new technologies into product field development as part of his roles in global science and technology and oversight of lab, greenhouse, and field research operations, having managed over 200 projects for clients, primarily focused on the performance of innovative and biological technologies. Before joining AgriThority in 2015, Ignacio positioned novel corn hybrids through countrywide testing, phenotyping technologies, and analytical tools, combining environmental data and crop models in his previous role as a research scientist at Pioneer Argentina SRL. Ignacio received an agronomy degree from the University of Buenos Aires in 1995 and attended graduate school at Purdue University and University of Illinois at Urbana-Champaign between 1999 and 2005. In between, he was a production manager in a 12,000-acre operation in Argentina. So with no further ado, I'd like to welcome from Argentina, Ignacio Colonna.
Thank you, Solito. I will be now sharing my screen. Can you see this correctly? Okay, good. So thanks, everyone. So today I will be presenting an overview of the use of digital images in field experiments. Thanks much for the American Chemical Society for this invitation. I'm honored to be presenting this to you. Hoping to present our motivation for being so involved with digital information in general. I will address that in a second. First, maybe a little bit of background of who we are to have a better understanding of where we come from in terms of using this technology. AgriThority is a consulting company. We work globally in projects involving product development, business development, and market development. Most, if not all, of our projects have to do with new technologies. A lot of these new technologies are on the bio side. Some of them are also chemical, but new chemical technologies, so well out of the ordinary. A key focus in my team in product development is how to develop protocols that are suitable for these new technologies. So there's a lot of constant learning on how to do things for these new technologies. We have incorporated digital tools into these protocols routinely for the past 5 years to minimize subjectivity. And I will address this very point at the beginning of my presentation now. And we have developed a system called TrialWorks for the past 3 years, which is specifically designed to produce reliable data out of large-scale experiments. And this model is heavily based on the use of digital information. And this is meant to be very different from your standard large-scale experiments in the sense of being as unbiased as you can get when dealing with large-scale trialing. Just briefly on my personal background on digital images, first experience in managing satellite and drone images was in 1997 when I was working as a farm production manager in Argentina. I was strongly focused in digital imagery during grad school at Purdue and University of Illinois at Urbana-Champaign. And then in Pioneer, we started using drone images for phenotyping, at least to attempt replace some of our ground-based phenotyping with digital images. And lately, in the last 80 years, we have been making intensive use of digital sources of information, not just images, as you will see in a minute, within AgriThority. So the outline of today is, first, I want to describe why are we so much involved into digital images or digital information in general. Then provide some background on digital images, just to make sure that we are all aligned into the jargon I will use later. I will then review the use of ground-based sensors, then aerial sensors, specifically drones, then satellites as source of digital images, and then I will provide some application examples. And then finally, we'll present a platform that we have launched very recently to help share the information with clients across the world. So first, I want to touch on the motivations for using digital information. I want to make clear where we come from. This is not a case in which we have a hammer, so everything looks like a nail. It's actually the opposite. We have lots of nails, and we were searching for the right hammer for the nails. So the reason for us, one key motivation for us was to come up with an objective assessment of the outcomes in our trials, and I will provide some examples in a minute. The second was to have a quality control tool for field trials. Then another motivation is the allowance for us and our clients for a continuous monitoring of crop growth during the season. You cannot get this by doing visits every week. It gets extremely expensive and also logistically very complex given our global nature. A comprehensive data collection in large-scale trials, and you will see what I mean when I address TrialWorks. This is a huge component of our TrialWorks program. And then finally, have a better tracking for our clients and ourselves of trial status during the year through online access of these images. So these are the 5 key motivations for us. Now, the first one is really the original one. This is why we started thinking about this. So a visual description of this comes from this 2 photographs. The above one is a typical view of a plot for a conventional chemical herbicide test. You see bare ground as a product of a commercial herbicide being applied in the plot. Everything is almost dead except a few weeds. And then you see the check around it that has no control. Everything is green. So here things are pretty clear cut. I mean, it's either dead or alive, no doubt. The herbicide works at maybe 90% control. Our challenge is the bottom one. We're dealing with a lot of trials in which what we see is something more like what you see there. Differences are a lot more subtle. It's not clear whether what we see is consistent, whether what we see is better or is worse, and then it comes to much of a subjective assessment. We are very commonly faced with the challenge of standing in the field with our client standing next to us, and all excited about what he sees in the treatments, and ourselves looking at the same crop saying, I don't see anything. And so it's very much depending on the viewer's perspective. So we don't want that to happen. We want to rely on objective assessment. So that's why we rely on things like this example here. And I'm going to address this in more detail, but this here you see on the left, the photo I showed you before, and then some seedlings coming from that trial that are not really very different. But then you see for exactly the same field, this is truly the same field, 3 satellite images with a specific spectral index, it's called NDVI. And from those images, we can compute the actual numbers for the trial, and then determine here in green the fact that the control treatment was consistently below the treated strips in the field, meaning that overall growth and overall greenness of the crop was actually lower in the control. And we can do this by using a quantitative tool coming from a digital image. So this is much more objective and much more precise than just walking the field and saying, hey, I think this looks better than this. And also we can do a lot of fine-tuning at a very precise scale. So this is what we want to get by using digital images. So now, just a little bit of a background to make sure that that the presentation is clear. First, what do we mean by visible spectral range and then near infrared? Maybe this is already very obvious to most of you. So the visible spectral range, of course, goes from the blue to the red in the spectral range. We make use mostly here of the absorbance in the blue and the red by the plant, and then the lower absorbance, higher reflectance of the green. And with that, we can make a few spectral indices that are very useful for quantifying vegetation growth. Then we also make use of what we call the near infrared, which is a spectral range that is just above the red, which is here, the near infrared. Most of what we use is on the shorter wavelength side, and the plants have a very, very strong reflectance in this side of the spectrum. Healthier plants have a stronger reflectance here. Plants that are close to being dead or with more senescent tissue have a lower reflectance. And then in some applications, we also make use of the thermal range of the spectrum, which may be useful sometimes for assessing crop stress. So this is kind of a basic components of the spectral range that we utilize. Now, what do we mean? So there's basically 2 types of images that we use when we divide them based on the spectral properties. The simplest ones are what we call the RGB. So images that only include red, green, and blue part of the spectrum, and just like the image you see here, including soybean seedlings taken with a simple camera phone. And maybe this is already clear for everyone, but just to be sure, each of the image you see here is composed of individual pixels, like you see here on the left, and each pixel has a value going from 0 to 255 for the red, for the green, and for the blue. And the combination of those 3 colors, 3 numbers, is what gives you the actual color that you see in the image. And so we can actually decompose this image into the 3 components, red, green, and blue, and see the distribution of those in a histogram, like you see here. And so with this, we can do a lot of manipulation of the image and come up with things that look like this, in which I can almost isolate the leaves from the background and actually work only on the leaves. I can actually cookie cut the leaves and make lots of operations on the leaf component of the image. Like here you see on the right, an image coming from a herbicide test in one field in South America, in which we have manipulated the color, the contrast, and the colors in general to highlight the weeds component and from the background. So with this, we can actually estimate the percent cover of the weeds per treatment. So the fact here I want to highlight is that there's a lot of manipulation you can do with the numbers in an image to isolate the physical components you want to work with. The other part, the other division that we work with when we divide things based on spectral properties is the multispectral images. And multispectral images include bands, not just for the red, green, and blue part of the spectrum, but also near infrared and sometimes also mid infrared. And I will show that in a minute. But basically, it's the same thing I showed before, but with more components to it. And each band, it's an individual layer of information. So some of them have values by themselves. Some of them have values in computing indices that are computed based on multiple bands. And then some of them have value using them, if some of you that are more used to the statistical world, using them in a kind of as a multivariate data set. And then, like I said before, some of the images include a thermal band. This is an example of a thermal band in a plot trial. The resolution for the thermal band is a lot coarser than for the near infrared and the visible components. So its use, it's a little bit more limited than the near infrared and visible bands. So now I will go step by step in the use of digital information. First, I will touch on the use of ground-based images. These are images taken, again, like with a normal camera or a phone camera for greenhouse studies, for field studies, and then for root analysis, which is something we do quite often. Then I will touch on ground-based sensors. These are still very common in small plots and sometimes for people that do field scouting. And then finally, I will address aerial images from drones and satellites. So first, how do we use digital images in greenhouse studies? These have proven quite useful for us turning a trial that usually has been based mostly on physical measurements into a more detailed assessment. In this case, we run a lot of trials for early development, for calibrating product compatibility, typically biological with chemical, typically evaluating of adequate rate ranges for a new product, and maybe a detailed study of mode of action for a new biostimulant. How does this biostimulant act on the plant? And so for this, we use a normal RGB image. And like I said before, we cookie cut the crop from the background. And from that, we can compute things like the leaf area along the time and compare treatments. In this case, you see a treatment that is the blue and one that is the green. And you see a very strong differentiation of treatments. And then we can also compute here what I have called the greenness. It's actually a spectral index, but quantifies the color of the leaves along the time of the experiment. And we can do that by individual seedling, not just for each image. And here I'm using an example of the software we use for that. We use a software here called ImageJ, which is actually an open source software. Everyone can use it. And we have developed some algorithms for doing that. But basically, it's coming up with a quantitative view of crop growth and can allow us a good assessment of plant of treatment effects. We can do the same thing with field photos. Well, anytime, so in my team, myself and a lot of my colleagues spend a lot of time visiting field trials, and we take a lot of photos. So this is an application of the photos beyond just showing to our client, hey, this is the field. And so we can actually turn the plant in the field, in the plot, into objects, and then compute measurements on those objects, just like we did in the greenhouse, but in the field. So this is another very good use. We can compute things like crop cover, what percent of the ground is covered by the crop, and also measurements related to the greenness of the crop. Third, we do a lot of work involving assessment of root growth. This is one of the most common claims in new technologies, mostly biostimulants and biofertilizer, have to do with increases in root growth. And a lot of the protocols are like pull the plant and take a look and score the root. But that most of the time doesn't work, because when you pull out a plant and you take a look, everything looks the same. So using a tool called WinRhizo, a software called WinRhizo, and scanner, we can estimate not just the overall length of the root system, we can also estimate the length of specific sections of the root, depending on the root diameter and the root hierarchy. You know, you have primary roots, secondary roots, nodal roots. So you can actually split the system according to the root section and measure the root length, root area, root volume, and a lot of properties from the root. So this is another use of digital images. You see here root length for the main root and for the secondary roots, again, for two different treatments, being significantly different for different rates of a particular biostimulant that we tested. And this is the same application, but used for root field samples. These are field samples that normally just remain as visual products, just someone saying, hey, this looks larger than this. Here, what we have applied WinRhizo to a field sample, and we were able to detect, for example, differences in insect damage to field samples for roots in large-scale experiments. So this is kind of a coarser approach than the previous one I showed, which was mostly based on greenhouse trials, and also provide a much better use than just looking at them and relying on such subjective assessment. So here are the plus and minuses of using ground-based images in your trials. First, they are low cost. So with almost any phone, you can take a good image. They help provide objective assessment in your trials, and also can provide useful material to the client, you know, beyond just the numerical data. The minuses is that you need to work on standardization, how you apply them. You know, there's a standardization on the distance, on the calibration for the image, for the camera. You need a systematic Q&A process for this, otherwise you can lose tons of hours filtering your images. And then the WinRhizo tool for roots, like I mentioned, is only as good as your root sample quality. If your sampling is poor, then your images are going to be poor. I will stop here for a minute. Yes.
We do have a question that came up here, and we'll.
Yep.
This is from Terence Mora. How do you account for spatial issues such as resolution and lighting when analyzing image data over large expanses, such as in the field you showed? We can back up if we need to do so.
Yes, this is a great question. Let me address this in a minute when I cover drone images. But this is a great point. Actually, it's one of the key points and one of the key quality issues we have when dealing with drone images. But for ground-based images, this is part of the standardization I meant. But yes, I will cover that. If I don't provide a good answer, please come back and come up with a question, and I will address it in more detail. So now going to ground-based sensors. One very popular sensors are NDVI sensors. Some of them are handheld, and then some other sensors like this one here are more like a pole with a camera that captures a specific radius in the field. So not just a leaf, but a section of the plot. We have used this a lot, and these are very useful. Generate a lot of data points per plot. The thing is, they are more intensive in labor than using a remote image, and also don't provide an actual view of the plot, just numerical data points. The other sensors that we have used and were very useful for us are static thermal sensors. These ones generate one data point every 15 minutes along all the season. And these have proven very good for us for assessing the effect of biostimulants in drought and heat stress trials. This is an example of a trial we run in the west of Argentina in a very hot and dry area. And with the thermal sensors, we're able to do quite fine discrimination of treatment effects on plant transpiration. So what are the plus and minuses of using this ground-based sensor? Again, very helpful for providing an objective assessment of trials. Some of them are very useful for scouting. You can bring them with you when you visit a field. So it goes better than just you writing notes, and provide very fine spatial resolution. Some of them temporal resolution as well. The minus, the QA process, the quality control can be, again, be very demanding. There's a lot of points that need to be deleted from a data set before going into analysis. There's a lot of device failure, data loss, a lot. We have dealt with that. And in tall crops, it is a challenge. If you're dealing in the subtropics with a 3-meter tall corn, you're limited to use these handheld devices.
Another question just popped up.
Yep.
That's from Pamela Breyer. I believe you have some case studies on upcoming for this particular question, and you mentioned some drought tolerance and heat stress as well. But moving forward with this in mind, do you think this all generates data that is more refined than needed, and whether these small differences that you mentioned translate into really important effects? So I think those are really good issues.
Good point.
As we continue to move forward.
Yes. So I guess the question here is whether the differences you see from a digital image translate into actual relevant differences in crop growth. That's a very good point. I will address that when I show an example of our use of digital images, including other measurements like final yield and other things, and show how those correlate and how we can use them, and when those are relevant and when we are not really sure. Yes, so I will address that in a minute. But yes, so the point is, learning about the correlation between what you see from a digital information source and the real world is a key component of the use of digital images. So there needs to be a calibration between your digital image and the physical world to ensure that you know what correspondence there is between a 5% difference in NDVI and plant height or plant biomass. So that's a key component, and we're working on projects related to that. So yeah, I will address that when I show an actual case. So now going to aerial digital images, what it is all about? It is all about converting a drone image into numbers. That's what it's all about. And a single snapshot of how we do it is, we place basically a grid on top of the image, and then we extract numbers from within that grid, and then we compute statistics based on those numbers. And in this case, we are showing an example of a treatment against a duty control for a specific trial in which a lower is better for whatever reason, and we see some difference between both. I will show this in more detail, but this is what it is all about. What options do we have here? First, we have low-cost options. Low cost, I'm talking about here a drone that is maybe around $2,000, and that most people running trials have, and a lot of farmers have, a lot of agronomist consultants have. These have improved very significantly in the last 5 years. When we started using them 10 years ago, they were no good at all for trials. The quality was very variable, very difficult to standardize. We lost a lot of time trying to come up with good data from this, but now they're getting much better. So this is an example of a low-cost, uncalibrated RGB image. So most of these low-cost are just RGB, not multispectral. What are they good for? They can provide several views of the trial along the season. So this is something very valuable. You know, anytime you visit a trial, you take an image with a drone. You can actually obtain simple green synthesis from this RGB. And for specific goals, these are actually quite useful. Depending on the trial goals, just the RGB-based index can be good. The minuses, the trade-off here is that sometimes if the image is not taken with proper care, the irregular shape in the image may make the process very time-consuming to fit it on your trial layout. The other one is that pixel values do not have a direct physical interpretation, and this maybe addresses partly the question that we just received. They are just numbers. They are linearly related to crop growth or to whatever disease incidence you're looking for. So you need to be careful with that. And finally, the brightness variation within an image can introduce a very significant bias. And I will show an example now about that in a minute. And then we have the professional drone options. These are much more expensive, and most of the time these are flown by licensed pilots. In certain countries you need an actual license, in other countries you don't. These are very fine spatial resolution. Some of them you can actually get to individual leaves, depending on the drone height. You can actually compute multiple response variables from a single image, depending on the band combination that you use. So you can see many different dimensions of the crop from a single image. What we call the ortho-directified image makes the process a lot more straightforward than what I mentioned about the warped images from the low-cost drones. Ortho rectification basically is about making your image look like the real world. The calibration, the spectral calibration, so calibrating against a true reflectance panel standard, can allow interpretation in real physical units of, for example, light reflectance. And then if you have large trials, then they become more inexpensive because cost gets diluted, you know, in a larger number of plots. What are the minuses? The cost may be very high for small trials. A lot of our trials are small and placed in many locations, so the cost is mostly in the travel, not in the image by itself. Usually this is between $500 and $2,000 per image for our trials around the world. The post-processing may be quite time-consuming many times, mostly the spectral calibration and what we call the stitching is not so much. I will show what I mean by stitching, but the calibration sometimes is time-consuming. And the thing is, if you are hiring an external provider for obtaining the images, the standardization of the quality control is critical. And it doesn't matter how professional the provider is, there is always a need on your end to have a QC well run, because there's many sources of errors, even if provided by the best professional pilot.
Ignacio, with the last two slides in consideration, we had a question from Caitlin Rim. Are there any low-cost drones that you can recommend, or are there any kind of hardware issues that somebody needs to take into consideration when they hire a professional?
Yes. Okay, that's a good point. Let me address that when I go step by step into the processing about drone options. First, I want to make sure that I clarify, I'm not the professional pilot expert, so I know drone brands and I can actually send some comments later, but I'm not the pilot expert. So the latest generation from the DJI drones that are the three generation from DJI is actually really good. So the Mavic Pro 3, I think it's called, is actually really good. but I can actually add that later to the presentation and then let you access that. But yeah, I don't want to claim to be a drone tool expert. I'm mostly expert on using the data later.
Quickly
and about
on the previous slide, Ignacio, a question from Aidan Roth would be about standardization of QC. What can you elaborate a little bit about QC standards?
Yes, let me address that going here. So
okay.
These are a few of the QC items, and this is maybe addressing also the previous question about what you need to take care of when working with an external service provider. So first, it's critical that you have georeference ground control points. And what we call a ground control point here is this square here, black and white, that can provide the actual location of a corner or a specific plot in your layout. Not having this generates hours or maybe a lot of time of you trying to overlay your layout to into the right position in the image, and also can generate some doubts about the overlaying across different times at different flights during the season. So this is really important. The other thing that can become quite relevant if what you're measuring actually has some relevance and we are trying to come up with, say, real reflectance values, is to have spectral calibration of your image. So here you see different panels with different tones of gray. This helps into the spectral into the spectral calibration of the image, so can provide much more quality into the indices you obtain. But out of these two, the actual physical location of the coordinates is really important. The other one is, and this may be really obvious, but validate your trial boundaries. We have tons of cases in which, for whatever reason, the service provider misses the actual trial and flies maybe the neighboring field. So make sure that your trial boundary actually corresponds to your trial. We have tons of cases in which, for many reasons, we come up with the wrong image and need to fly it again. The other point is the stitching. So the stitching, and this is for everyone to realize, a single image actually is composed of hundreds of individual captures by the drone. These captures are overlaid one onto the other in about 80%. So this almost an 80% overlay between one image and the next. And this produces what we call the orthomosaic, which is this. So this image here comes from hundreds of individual images. So this is very heavy in terms of the size of the files. And this is pretty automatic today. There's software that does this almost by itself. So there's not a lot of human participation in the process. And then a major component here is how you buffer your image to make sure that what you keep is actually really proper to the plot you are dealing with and not affected by the neighboring plots or fields. And this comes to the also the light heterogeneity question that had been given. So what we do is for each plot, we impose what we call a buffer area, and we analyze only the central area of the plot. This is an example for small plots, and this on the right here is an example for large strip trials. In large strip trials, we generate a subgrid, and this subgrid includes dozens of data points within each large strip. And finally, the fifth step is we generate many spectral indices and run secondary analysis, and then export values to run statistical analysis for this. So what are some of the quality check watch-outs? And this addresses directly the questions that I received a few minutes ago. This is one key issue that we have that is not obvious, which is the contamination of your image from neighboring plots, from neighboring fields. In this case, you see an image from a trial that is completely affected by the neighboring field that has a bare residue. So some of the plots have to be discarded. So this needs to be inspected visually with a lot of care. The other one is, one of the very common things we do is we classify image into crop and non-crop areas. And this, you do need to do a fine scale check and make sure that what you're classifying as crop is actually a crop. Do not rely on automatic algorithms without checking them. We have many cases in which this does not work really well. So a careful choice of your spectral index that you use to actually feed your algorithm is critical. The algorithm may be the same, but you can feed different spectral indices for this. Make sure you check this is correctly classified. The other one is plant counts. A lot of the use for images today is to run plant counts. This works well for row crops like corn and sunflower. It does not work well in general for soybeans or wheat or any very dense crop. And this is especially difficult when you have background such as that in no-till fields. No-till fields are quite complicated, so make sure that you do a very detailed inspection of what the algorithm classifies as a plant in this case. This is one major caveat. So now going into some examples of, I will provide several examples how things look once you have run the analysis. So this is a corn seed treatment study. And this is an example of 4 different outcomes that we produce. And this, you see a large strip. Each strip is divided into subplots within the grid. And here we actually estimated plants per square meter. And you see here, there's quite a variation here, going from 5.6 to 8 plants per square meter. Percent crop cover, a good indication of early vigor. An index here that is called mean excess green. Excess green is the index name, and then another index that is called vary. Both are indicators of overall crop greenness, and they have been quite valuable for us. Another thing is that many times we can compute not just the mean spectral index on the plot, but also on the specific area of the crop, not just the plot, but on the crop. Another application here is the use of images, historical satellite images for field zoning. We want to make sure that we install a trial in a homogeneous area. So we do long-term analysis of images to make sure that we don't lay our trial on an area that had been affected by a flood, for example, or by soil heterogeneity. This is an example of the use of such an approach for classifying a field into zones. I will not get into detail, just these are different layers of information, all of them coming from digital sources. And with this, using a clustering algorithm, we classify the field into different areas. And using those areas, we run our, in this case, yield response analysis within that areas. And we have seen consistently is that responses can be very different in one area versus the others. And this can tell us a lot about the product. Here, I'm showing analysis of yield. This can be applied to other measurements, biomass, height, plant counts. But the fact is that the zoning approach is a lot more revealing than the no zoning approach. There's a lot included in the large field that you can reveal by splitting the field into parts. Another example here is a herbicide study. This is a macro plot study. These are about 10 meters by 2 meters, well, almost small plot. We use low height RGB images. I think these are half a centimeter in resolution, if I remember correctly. And we run here a spectral index. We can classify each plot in percent cover by weeds, and then analyze the weed cover as a response. And this has proven very, very strong for analyzing some of the images in complex situations. Clients are very happy with this, both because of the visible, but also because of the quantitative use. And then we can turn data into numbers, like I said, and in this case, it has helped us understand the effect of a biofertilizer using percent crop cover, which was not obvious at all from the naked eye, as related to initial soil nitrogen content. So using the crop cover was much more, much more detailed than what we could capture by measuring things like crop height. Another example here is the use of digital images in Brazil for soybean disease assessment. We have compared here, and this is an example of the question that someone had: how does this relate to real world? We have compared the visual scores of crop severity and incidence with the digital assessment, and we have proven that at which point we can start using the digital images. We could not go very early in the crop, but we were very effective in differentiating treatments, both in terms of crop cover and greenness in the plant. Another example here, and this is especially meaningful, is taking weekly images from satellites. These are high-resolution satellites at 3 meter. It's called a planetscope constellation. And so what we do here, we compare the relative NDVI between treatment and control across the season. And here you see a peak meaning higher value for the treatment. And we correlate this with the rainfall events during the season. And what we observe here is that right after a very intense rainfall event, we see a peak in the treatment effects. And this made a lot of sense in this case, in which we were treating a product that was aimed at improving soil infiltration of water in fields that were affected by salt. So this increase right after the intense rainfall season was particularly meaningful in understanding the end of season results in our trials. And finally, I wanted to share here the platform that we have developed with a company called Altea for sharing our information online. This helps a lot in making the process a lot more automated. Before, we had a lot of manual work in desktop GIS software. And just addressing the question that someone had before, a lot of the early QC process is done in a software that's called QGIS. It's very popular. And now we still rely on that software for some very specific projects, but a lot of what we do is within this system now. So here we can host images for every project. We can allow client access to specific images, and we can run a lot of the processes in an automated fashion with a proper QC at the right steps, like I showed before. And here you see you can lay your plots, and some of the plots you see here look really weird because they have specific issues that make us the need to break them out and to make sure that we don't capture things that are heterogeneous in the field. And we can host a lot of different images in the same site. So, just reviewing what I said before, our key motivations here are first to have an objective assessment of crop growth versus the visual scores. We are now working on a specific project for a client that wants to replace ground scoring in its phenotyping program for corn breeding, for early vigor, plant health, and pre-harvest stock lodging, basing that on digital images. So we are developing, hoping to develop a model that can predict those with little sampling from the ground. Then we use them for quality control. For example, mapping heterogeneity in the field, that is very critical for us. Like I showed in the example of the different zones within a field. Then we monitor crop growth across the season, like I showed in the example for the biostimulant, correlating with rainfall during the season. Comprehensive data collection in large-scale trials, like I showed the example of plant counts. We can count plants in an entire field instead of just relying on a few data points. We just need to make sure that we identify plants correctly. But this is a huge change for large strip trials. And then finally, like I showed in the application, client remote tracking of trial status in almost real time. Delivery time is typically about a week. So within a week, client can actually watch the status of the trial. So finally, in case all of these was too hard, this is a kind of a more a funnier application, which is, I don't know how many of you are into soccer. Last year we won the Global Soccer Cup in Argentina, and some Argentinian fans generated the face of our soccer star Lionel Messi in the field by changing the plant density for corn. And they captured that with drone images and satellite images, and this made the news in CNN and Reuters and others. So this was a kind of a fun example of the use of digital images for non-agricultural uses. So with this, I will now stop, and if there's questions, I will address more specifically.
Fantastic. Thank you so much, Ignacio. Now, we do have some time for questions, so please keep them rolling in. The first one that rolled in on the latter part of the presentation, Ignacio, would be an explanation and elaboration on the calibration and QC processes of thermal imagery.
Yes. Let me show a couple of examples here. Let me go back to let me go here. Yeah, so the calibration, so this is a process that again is much more on the side of the professional drone provider side than on our side. What we do is that we quality check the calibration. So there's a couple of things we do to make sure that things are correctly calibrated. One of the things is we compute some key spectral indices like NDVI and others that are called NDRE, for example, and we make sure that the values for these indices are correlate properly with different areas with different levels of crop growth and areas that have or don't have crop. Many times, a wrong calibration, a wrong spectral calibration of the sensor, of the image actually, implies that areas with larger crop growth actually show lower spectral index values, and the opposite, which is exactly the opposite of what should happen. We had that many times. So correlating your spectral index values with areas of different crop growth is one good step to take. Also, calibrating, checking your index versus this, what I showed here, versus crop and non-crop spots, very important. Sometimes there's weird things that happen and make your shadow areas peak in your index values. So that's one key thing. Then, like I said, there's more obvious calibration issues, which are more like the physical calibration, which is making sure that your image overlays correctly with your grid. Sometimes there's some deformation in the image that generate a non-linear overlapping. So maybe your plots over here will overlap correctly, but your plots over here will not. So don't just go the quick way. Make sure that every plot correctly overlays with your image. So I guess those are two comments that are one part of the critical thing that we check. I'm trying to think if there's anything major that I have missed here. Yeah, and then like I said before, run a couple of algorithms. Like I said, in this case, the plant recognition algorithm. If the plants are all poorly recognized, this is an indication that there's something maybe wrong with your image calibration. There may something that may not be allowing you to separate green from background.
Pablo Casas, on top of congratulating Argentina for the World Cup, he had a question on Sentinel-2 imagery. Are you working with Sentinel-2 imagery? And then for extensive crops, is high quality imagery a must?
Sorry, can you repeat the second one, Solito?
Yeah. Let's see. First is the working with Sentinel-2 imagery, and then the second one would be for extensive crops or high quality imagery. Is that a must for?
Okay, I understand.
And we might ask Pablo to elaborate on that second part if we don't get that answer.
Yeah, good question. So we do work with Sentinel images, mostly for uses like the one I'm showing here, to get historical views of a field. We can get maybe 5 years of NDVI for a specific field and allow us to understand variation across wet and dry years and classify that for placing a trial. We are not using Sentinel for our actual trial data gathering. The thing is, Sentinel has a 10-meter resolution right now, and the problem is that most of our trials involve strip widths that are larger than 10 meters, but not too far. And there's a lot of what I call, remember what I explained as the spectral contamination, things coming from the neighboring fields affecting your plot. That happens when you have too wide of a pixel size. So Sentinel is good for us for uses like this. We use it for very, very large strip field scale trials, but most of our trials that are like this one here, strips, we rely on PlanetScope, which is 3 meter. There's one that's called SkySat, which is 50 centimeter. It is more expensive, a lot better resolution. In terms of quality, I would say that for a strip that is like ours, most of the time not wider than 20 meters, the PlanetScope is a no. There's no way you can make a good use of data in a 20-meter strip coming from a 10-meter pixel. There's a lot of influence from the neighboring strips, but we can, yes, use Planet and, of course, SkySat. Planet is a lot cheaper, and then you can download images from the past season. SkySat, you need to plan ahead. You need to actually contract that most of the time ahead of the season, because otherwise you just need to be lucky and get images that has been taken in your field.
And as a reminder to the folks, we will catalog all these questions. So we are about 3 minutes away from the top of the hour. So I just wanted to let you know that we will be able to, we're in a position to answer follow-up questions after the presentation. Terrence Mora asked about how sensitive imaging technology is with respect to plant diseases, such as diseases with less contrast than soybean rust or wheat powder mildew, and what platform do you recommend for those?
Yeah, this is a good point. One thing is we have not ever focused on identifying specific diseases. We have focused only on assessing overall plant health and then ground validating which was the disease being present in the ground. So that's one thing. We have never actually identified specific diseases. As for sensitivity, using multispectral images is much, much better than using RGB for diseases. And like the example I showed here, we have been able to detect diseases in, mostly we have worked in soybeans at relatively advanced stages. In soybeans, one of the limitations is a lot of the disease characterization goes below the crop top of the canopy. So sometimes an observer can actually score a canopy under the top and detect something, but the drone can only detect things on the top. So you see here, the visual score started separating itself from the treatment at earlier stages than with the drone. So the drone at the same stage was still at parity, and we were only able to detect the differences when the crop was started to senesce. So the point is, you may not be able to detect diseases very early in the season with a drone, but once crops start to yellow, then the treatment separation becomes much more evident. And again, multispectral would be the recommended way to work here.
I think we have time for one more from Pablo. There are a lot of vegetation indices. Which are you using for rice trials? And you might be able to expand on this even beyond rice. Are there differing vegetation indices for crop growth stages as well?
Yeah, this is a good point. So for early in the season, when we want to identify crop from background, usually we rely on RGB images, which usually have a better resolution. And most of the time there we use excess green. So that has been very useful for us for any crop. Now, later in the season, we use most of the time NDRE. Now for rice, it's especially challenging. And here we have been experimenting with different algorithms, different indices. I think we were quite successful in still using RGB for rice. Excess green has been quite useful, and NDRE as well. There are some completely out of standard algorithms developed for rice that we have also tested. But I would say that NDRE and excess green are one of the most commonly useful for us.
All right. Well, I'm going to wrestle control the screen so we can close this out. Again, we have all the questions cataloged, so we're certainly in a position to follow up with our audience today with answers to those questions. But let me share the screen here. Yeah, thank you so much to you, the audience members, for your participation in this webinar. I'd like to thank our speaker, Ignacio Colonna. He's been fantastic today. Europeans Agroscience Services, our sponsor, and our webinar coordinator, Kyle Sawyer, and the webinar committee for their work in putting together this presentation. You may be asked to complete a follow-up survey after the webinar, and we look forward to your feedback if that's the case. Future webinar planning is ongoing, so please contact me if you're interested in supporting our efforts to publicize the series and to contribute to future content. You're the content providers, you are the audience. So thank you for your attention, and we'll see you next time on May 17 at noon Eastern in the US for Susie Banzinger's webinar on recruiting talent in the agrochemical business. We do get this question a lot. You can find our recordings of past webinars at www.agrodiv.org/webinars or the Agro Division channel on YouTube, as you can see right there. So on behalf of the Agro Division, on behalf of Ignacio, and on behalf of Kyle, have a great afternoon and have a productive week.